Okay, so good morning to everybody and very welcome to this matrix biology course that this year is a joint event with the DICU Summer School in Fibrosis. Today, you will be introduced to the extracellular matrix. You will learn about imaging in biomedical research and about proteoglycans. Our first speaker today is Professor Donald Gullberg. He's from the University of Bergen, and he's also a co-organizer of this course. He has today 2 lectures, and he will first introduce you to the extracellular matrix, and then afterward talk about integrins. And I want to say that the audience is muted during the talks, but you can ask questions after the talks, either by using the raise hand or the chat function. And with this, I will just give the word to Donald, please. We look forward to your lecture.
Thank you very much. So I will then just join Marion in welcoming you to this course. I think we have a week full with exciting talks, and I think you should be aware that it's we have collected quite a good mixture of speakers. It's new to us, of course, to have this course digital, so we just hope that everything will work, especially the part where we have recorded the practical moments of the course. Okay, so I will start today with kind of a crash course in extracellular matrix and integrins. And for some of you, this will be old news, but so this is meant to be like an overview. And we start with extracellular matrix. And this is just an outline, what I will be talking about. We'll be talking about different types of ECM molecules, collagen, proteoglycans, and a very big group called multi-adhesive glycoproteins, which include some favorite molecules, laminins, fibronectin, and towards the end we just say some general words about the function of the ECM. So generally when okay. So Maureen is saying we can't see PowerPoint. So is it visible now? Torsten?
No, it's not there.
So what's
Just
the problem?
share it again.
Yeah, okay. I have to go. So shall I close it or share screen?
Just click on share screen again.
Yeah. And then Microsoft PowerPoint, share.
And the presentation. Yeah, now it's there.
Okay. And then I go to present review. So it's okay now? Yeah,
yes.
Okay. Sorry. So it had been on for too long. So this is the outline of the talk. So in general, when I talk about how cells receive information from the surroundings, I divide the signals that a cell communicate with its environment into 3 parts. Soluble factors like growth factors, cell-cell interactions mediated via cadherin and cell adhesion molecules, and the third type of information collection that the cell pursues constantly is that with the extracellular matrix. An extracellular matrix from now on, as you see, is abbreviated ECM, right? And this is just a schematic illustration of these different types of interactions, cell-cell contact, soluble interactions, and what we will be talking about for the next 2 hours, the interaction with the extracellular matrix that occurs at very specific site via very specific receptors. And I usually also tell my students that when I started in this field a very long time ago, then the dogma was that cells did not interact specifically with extracellular matrix. That the extracellular matrix was just something diffuse that surrounded cells, but at that time, no receptors had been purified for the matrix. So that's the big difference today from what it used to be like. And this is a schematic illustration how we can think that it looks at the surface of a cell, and we've zoomed in. We see the phosphobilayer, we see the cell surface receptors, a group of molecules called integrins, and we see the extracellular matrix. And if you look at what kind of molecules are present in this matrix, I would think that we're maybe looking at the fibroblast. Since we're seeing fibrillar collagen, we see a protoglycan, we're seeing an abundant matrix protein called fibronectin. Whereas a molecule that whoever made this graphic illustration, the laminins are usually not present in this matrix, is present in basement membrane. And these ECM proteins then are synthesized in the cells, secreted, and different cell types contribute to the matrix. And so nowadays we have sequenced the genome, right? We know how many proteins we have, and we also have categorized the proteins that belong to the extracellular matrix. And there are a large number of proteins. So to make some sense of all these different proteins, we sometimes divide them into 3 categories: collagens, protoglycans, and all the rest, which are many, many proteins. We sometimes, in lack of other good terms, call them multi-adhesive glycoproteins. So they're all glycoproteins, and multi-adhesive refers to that they interact with each other and with cells. Some of them are not so, do not interact so well with cells, but in general, they interact with each other. And today we don't have time to go through all these proteins, but we will just mention some of the main players like fibronectin, laminins, nitrogens, and yeah, we will also mention some of the other matrix proteins. So on top of categorizing proteins into 2 categories, we also sometimes distinguish between 2 different types of extracellular matrix. One we call the interstitial extracellular matrix, and that is a matrix that is rich in fibrillar collagens and typically contains a fair amount of protoglycans and other glycoproteins. So the skin, the dermis is an example of interstitial extracellular matrix. Bone, cartilage, tendons, they're all examples of tissues rich in interstitial extracellular matrix. The other type of principal type of extracellular matrix is a basement membrane or basal lamina, which is another name. Which is a thin network of which is composed of actually 2 networks, as we will see, a collagen type 4 network and a laminin network, and together they form the basement membrane. A basement membrane are then the thin network which underline all epithelia and endothelia. And like a stocking, it surrounds all muscle, skeletal, cardiac, and smooth muscle. And it also surrounds adipocytes and Schwann cells. The basal membrane has tissue-specific functions. In kidney especially, it's important for filtering, but in most tissues, it's there to give stability to the tissue. And this becomes very obvious when you have genetic diseases affecting basement membrane in skin and muscle, then you have very drastic effects in these tissues. This is a schematic illustration, which is not super duper particular good. Basement membrane here look more diffuse than what they are in real life. Basement membrane are, as I said, thin sheets, so they're usually more discrete than in this graphical illustration. And here you see skeletal muscle, muscle fiber, that is surrounded by basement membrane, whereas here in an epithelial sheet, it's at the basal surface of the epithelial cells. So if you look at most tissues in the body, with a few exceptions like the brain, for example, you will see that it contains a mixture of interstitial matrix and basement membrane. So here we're looking at the gut. Here these epithelial sheet will sit on a basement membrane. Here we come into a section where we have an interstitial matrix. Here again, we have two layers of smooth muscle cells where each individual muscle cell is surrounded by a basement membrane. Here again, interstitial matrix. And here again, we will have a thin basement membrane rich in laminins and collagen type 4. Here is another example of where we have the two types of matrix. This is a cross-section of the skin. Here the keratinocytes sit on a basement membrane, and if we go down into the dermis, we have an interstitial matrix rich in fibrillar collagen, and if we zoom in on the fibroblast, the cell is anchored via integrin to fibrillar collagen. Right, so then that was background, matrix, how we divide it, and now we will very briefly go through some of the major components in the extracellular matrix, and we start with collagens. So you would think that defining a protein like collagen is easy, but actually for various reasons that is not the case. But we will not go into detail. Suffice to say that we define a collagen as a protein which has one or several domains which are composed of a collagen triple helix. And the second criteria that must be fulfilled, this protein must be a structural protein. So it must give some structure. And the reason why this criteria is included is because there are proteins which have a collagen triple helix as the structural building block, but they are not part of the matrix. So we can say a lot about collagens, and we could have 5 lectures just about collagens, but we will go through it very quickly. So collagens are the most abundant vertebrates, abundant protein in vertebrates. That is because collagen makes up the skeleton, and the skeleton is a big part of us. So collagens are part of skeleton, cartilage, and tendon. Now, when we have sequenced the genome, we have with the current definition, where the collagen family has 28 members, and it will probably stay at that. But it is, we know the total number of protein, but it's a matter of definition. Some proteins there are disagreements on, but most likely there will, in vertebrates, higher vertebrates, it will be 28 different. So the collagens can then be divided into many different subfamilies. but maybe the two major ones is the fibrillar collagens, which are the most abundant collagens, the collagens in bone, cartilage, and skin, etc., and network forming collagens, which includes collagen type 4, which makes up the basement membrane, which we have throughout the body. Just a few words about the collagen triple helix. So the collagen triple helix is composed of 3 individual chains. And these chains can all be the same, then we have a homotrimer, or they're different, then we have a heterotrimer. And it can be 3 alpha 1s, 1 alpha 1, and 1 alpha 2, and 1 alpha 3, and so on. But in order for a collagen triple helix to form, there are some criteria that must be fulfilled. First, every third amino acid needs to be a glycine. And second, we must have hydroxylated proline in this collagen domain. And here we see the triple helix. 3 in this case, we have an alpha 1, alpha 2, and alpha 3 there with different colors. And here, this slide summarizes why every third amino acid needs to be a glycine, because glycine faces inward in the triple helix. And the way it's packaged, there is not room for any side chains. Whereas the proline side chain, which is a bulky side chain, is on the outside. And why it's super duper important that collagen is hydroxylated. This is because this leads to the formation of hydrogen bonds that keep the 3 chains together. So collagens are then a bit special in many respects. So protein synthesis is not like your average protein. So we'll just say a few words about synthesis. So collagen is made in the ER, and then it's post-translationally modified both in ER and in the Golgi, and then collagen is secreted. But at the ends of the collagen, once it is secreted, there are large globular amino and carboxy propeptides, and these needs to be cleaved away for collagen fibril to form, and these then assemble into fibers. Okay. And this is a schematic illustration of the synthesis. Here you see the hydroxylation, and in the Golgi we have glycosylation. Here the collagen is secreted. Here the propeptides are cleaved off. The mature collagen is called tropocollagen, which forms a fibril and then assembles further into a fiber. And this is another schematic illustration of fibrillar collagen. Okay, that was super short about collagen. Just a few words about collagen diseases. So the genes for the individual alpha chains are large. So there is ample room for mutations, but these mutations are relatively rare still. But the diseases that you are affected by are put into different categories. So one is the Ehlers-Danlos syndromes, which is now more than 10 different types where you have mutations in different collagens and different proteins that associate with collagens. And characteristic of this is that the extracellular matrix in the skin then is very elastic. And I think when I updated this lecture recently, I put into an updated sub diversification of the Ehlers-Danlos classification, where you have a classical, which is due to, in the majority of cases, to mutation of collagen type 5. And then there are some more recent proteins, a protein called tenascin X. And here again, the different genes mutated in different forms are listed. Another disease that is caused by collagen mutation is osteogenesis imperfecta, which is mutations in collagen type 1, the collagen that exists in bone. And depending where the mutation is, the severity varies. So the three collagen chains come together from the carboxy terminus. So if you have mutation in the beginning, that's more severe because that can interfere with assembly of collagen. So another point I want to make is that if you work with collagen, you should of course know the history. And part of collagen history is of course that scurvy is caused by defects in collagen synthesis. And we will just say a few words about this. So I said before that collagen hydroxylation is important for the stability of the triple helix. And the enzymes that hydroxylates proline needs vitamin C as a cofactor. Just a few words about scurvy. So, as everybody knows, scurvy affected sailors, and for a long time one didn't know what why they started to bruise easily. If they hurt themselves, their wounds didn't heal. They started to have bleeding gums. The periodontal ligaments were affected, and until one discovered that this was due to the lack of citrus fruits. And at that time, the protein collagen, of course, wasn't known, but now understanding collagen biosynthesis, we can understand the underlying reasons for the disease. And in order to understand that, you also need to understand that collagen is a very stable protein. In many tissues, the turnover rate is very low. So once you've stopped growing, like in most sailors, they've stopped growing, then the collagen turnover in the bone is not affected. But at places where collagen turnover is high, like in the periodontal ligament in the teeth, every time you chew, you produce new collagen. It's constantly being produced in the periodontal ligament. And when you have a wound, then a new collagen will be produced to close the wounds. So all these places where there is a turnover of collagen are those that are affected if you have a problem with collagen synthesis. And this coincides with the symptoms of scurvy. So the enzyme that does the hydroxylation is called prolyl hydroxylase, and there are different prolyl hydroxylases. And the enzyme needs iron and vitamin C, and to put on this OH group on the proline. So for normal collagen is a stable protein, and it's not denatured at body temperature, of course, then it wouldn't work. But if you produce collagen without hydroxyproline, it's much more stable. It denatures at low temperatures. So in practice, if you have this mutation in cell, the collagen would never be secreted. It would never form a stable triple helix inside the cell at 37 degrees, right? After collagen is secreted, it's cross-linked. And this is something that's important as we age and our skin becomes less elastic. In the context of tumors where we have a stiff matrix, you're a higher chance to develop breast cancer if your breast matrix is stiff, and the stiffness in tumors in itself is a risk factor that propagates the disease. We will not mention so much of this, but in different pathological conditions, you also have modifications of the collagen. Specific amino acids can be modified, and this changes the properties of the protein also when it comes to interaction with cells. And especially amino acid arginine can be what's called citrulinated, and this affects how, for example, integrins recognize the collagen. We will not go into so much detail here, but that that has implications for inflammatory diseases, for example, when cells will behave differently when collagen has been post-translationally modified. So collagen is, as I said, everywhere. This is from a visit that I made in Shanghai, and right outside our hotel, we had this big sign of collagen. And if you go to any health store in the US, collagen will be everywhere. So last time Marin and I was in San Francisco, we could even get collagen water when we went into the nearest grocery store. So collagen has become very popular to take as pills and to have it supplemented in the water. And in Norway, of course, you can buy fish collagen. And if you eat these tablets, your wrinkles are supposed to go away. However, this does not work. So collagen, when you take it in orally, is degraded into individual amino acids. And individual amino acids do not know that they should go to the skin and make your wrinkles disappear to make new collagen. So all fake. The collagen story is much more complex than what I'm telling here. Collagen type 1 doesn't form fibril by itself. It forms kind of alloys with different collagen. So it's usually collagen type 1, 3, and 5 that together combine and form some sort of fibril. And collagen type 5 is usually at the center of this alloy, and is only the termini of this collagen type 5 core that's sticking out of these complex heterotypic fibrils. This is a picture from the skin where you can see a cell surrounded by collagen in different directions. So that was fibrillar collagens. We now move to type 4 collagen, which is a network forming collagen. So one huge difference between collagen type 4 and collagen type 1 is how sensitive the different collagens are to proteolysis. So collagen type 1 is among the most stable proteins that there is. So only specific collagenases can break it down, but it's resistant. It's made to be a structural protein. But collagen type 4 has a triple helix that is interrupted by non-triple helical places 23 times. So 23 times along the triple helix, there is chance for proteases to cleave. So a collagen type 4 network is sensitive to proteolysis. This is a slide showing the other types or subtypes of collagen. We will not have time to go through these. In this slide, we can see some other proteins that have a collagenous domain, but they're not called a collagen, usually because they're part of the blood and they're not structural proteins in the extracellular matrix. So they don't fulfill all the criteria to be a collagen. So to summarize of collagens, they give mechanical stability, restrict stretching forces, and usually when you have a mutation in these, the disease is manifested in the tissue where the collagen is expressed. So if something is expressed in bone, then you will have a bone phenotype. If it's in cartilage, it's a cartilage phenotype. So this goes for osteogenesis imperfecta, chondrodysplasias. If something is expressed in muscle, it will give a muscle phenotype. And this is collagen type 6, which gives Bethlehem's myopathy. Collagen type 10 is in cartilage and also gives a chondrodysplasia and so on. So again, collagens gives mechanical stability and different collagens in different tissues. I think we will not go so much into defects, but you can understand the function of protein by studying human mutation, but you can also study mouse mutations. So this is the summary of the phenotypes of the different collagen knockouts. Maybe we should just mention that if collagen type 5, which is a minor collagen, is mutated, you have no fibrillar matrix in the embryo at all. So based on this, it has been postulated that collagen type 5 is a nucleator important for the fibril formation of all fibrillar collagens. And this is an area of intense research. We probably not go so much into this either, that if you have mutations in individual collagens, collagen binding integrins, this gives a mild phenotype indicating that collagen binding integrins have a role in processes other than embryonic development. And we will not go so much. Pardon me?
1 more minute. Oh, sorry.
No,
Sorry. No.
We will not go so much into also that the fibrillar forms of collagens are so tightly packed that in mature tissues, integrins are not able to bind. And we have instead introduced the term column-breeze for collagen integrin bridging molecules, molecules like fibronectin that bind to collagen. So in the tissues, cells interact with collagen either directly via collagen binding integrin or indirectly via proteins that in turn then bind to collagen. And this a very high profile paper showed that when you extracted collagen fibrils from tissues, cells didn't bind, which was surprising. But this is probably due to this, that in mature tissues, the collagen fibrils are coated with proteins and are so tightly packed that integrins don't have access. So this is not completely understood, but it's thought that in mature tissues, there is limited availability of integrins to collagens, and it's more in situations where collagen is newly produced, you have a high turnover, like in fibrosis, that we will talk much more about in this course, or in tumor stroma, which is another situation where you have high turnover of collagen. And just recently, it's becoming also a hot topic to understand how collagen is actually assembled at the cell surface. That although collagen can self-assemble, this is why you can make collagen gels in a test tube, as we will do in the practical part. But there is a certain advantage of cells helping. You can then direct the assembly of collagen. Okay, I'm running out of time, so we have to speed up a little bit. So you will hear more from Marion about glucosaminoglycans, which are composed of unbranched sugars, usually sulfated, and can be divided into different groups: heparin, heparan sulfate, keratin sulfate, chondroitin sulfate, and hyaluronan. And a protoglycan is composed of a protein part where you have this glycosaminoglycan covalently attached via a serogly region. And this is an example of the cartilage protoglycan called aggrecan that form large complexes where you have the glycosaminoglycan chains, which are then interacts with hyaluronan to form large complexes like here. And in the cartilage, this complex binds a lot of water and fills the purpose of withstanding pressure. Just as we said that collagen is found in health stores, so is protoglycans in the form of usually shark cartilage or other forms that are sold. And you are made believe that if you eat this, your joints will feel better. But as the same way as with collagen, all fake. If you take protoglycans orally, they will be degraded, and there is no way in the stomach that the degraded components know that they will go and make new cartilage. One use of protoglycans of glucosaminoglycan is hyaluronan, which is used in eye surgery. It's very viscous. And Restylane is hyaluronan based. Still not in the market, but it's possible that, and developers are discussing that in the future you will be able to buy your ready-made six-pack. Protoglycans are also some type of protoglycans exist on the cell surface where they sit and bind growth factors and present them to the growth factor receptors. And you will hear more about this in the course. So protoglycans endow tissues with special properties, store growth factors, act as co-receptors for growth factors, and a special protoglycan called heparin is needed for mast cell function. And we said that protoglycans contain large amount of sulfate groups, bind large amounts of water, and resist compression forces. And this is a summary slide of this. Okay, so we have 6 more minutes, correct? Then we'll very briefly say a few words about laminins and fibronectin. So basement membranes are composed of two networks, a collagen type 4 network and a laminin network. And in between is nitrogens. And also in these networks are protoglycans. So the laminin network forms by non-covalent calcium-dependent association. So if you remove calcium with EDTA, this network will become loose. So you see here the laminin molecule, which are cruciform molecules. And the other network here in the red is a protease-sensitive collagen type 4 network. Here is the schematic molecules of laminin molecule, which are large molecules composed of alpha, beta, and gamma chains. And it's the carboxy terminus that contains the integrin binding sites. And recently, this binding site has been mapped into the gamma 1 chain of one laminin isoform. And in basement membrane, then here is in the hemidesmosomes, cells via integrins bind to a special laminin that's in the skin basement membrane. And the basement membrane in turn is anchored to the underlying interstitial tissue. If you have mutations in laminins in tissues where you're dependent on mechanical strength, you will have defects in the tissue structure. So this is a skin disease called epidermolysis bullosa. And there is also other muscle you can have if you have laminin containing the alpha 2 chain, you have defects in skeletal and cardiac muscle. The last protein we will say a few words about is protein called fibronectin that all of us have in blood. It's produced in the liver by hepatocytes. We have a fairly high concentration of 200 microgram per ml in serum. And but fibronectin also exists in tissues during development and at times of tissue injury. And fibronectin is easy to purify because it's also present in blood and has been very important when receptors for integrins were isolated. And the magical amino acid sequence that fibronectin binding integrins bind to is RGD, which stands for arginine, glycine, aspartic acid. And this sequence is present on the loop on the fibronectin type 3 repeat. And it's very strategical, easy for integrins to bind to. And fibronectin is a dimer to only one gene, which can be alternatively spliced. The subunits are disulfide bonded, and then this cell binding sequence is present in the 10th fibronectin type 3 repeat. And fibronectin is then used in vitro studies of cell migration. So since cells have multiple integrin receptors for fibronectin, they usually like fibronectin. And if you put a stripe of fibronectin on a glass, cells will migrate on this stripe. In the tumor microenvironment, there is an increased interest in understanding the extracellular matrix. What is collagen doing? How can collagen create highways for migrating cancer cells? How can the extracellular matrix create niches that the metastasizing cells go and find and decide where they're going to form the metastasis? And it's also realized, of course, that in the tumors, the extracellular makes a barrier and also is involved in the development of chemoresistance. Here is a summary slide from a book chapter we wrote where we see the structure of some of the major components we talked about today. Here is another summary slide showing where the function in the context of tumors of some of these proteins in the forming pre-metastatic niches and affecting cell migration and so on. And one protein that's been especially interesting in the formation of a metastatic niche, that is that cells go to place in the body and deposit tenascin C that the tumor cell will then find, is the tenascin C, a large molecule, and in this case the bone marrow cells that go there first and condition the pre-metastatic niche. So with that, my time is up, and the last 30 seconds, we can look at this summary of the role of the ECM. So the ECM is above all, and very importantly, a mechanical support. But with time, we realized it has many more functions. It is a reservoir for soluble factors. It promotes cell migration, promotes cell polarization, affect differentiation, prevent apoptosis, and what we will talk about in next lecture, it binds to receptors to generate signals that are important for many things. With that, I stop. Thank you.
Thank you very much, Donald. Do we have any questions here? I would encourage, especially also the students here or the PhDs, that you should take the opportunity to ask. There's a question here for you, Donald. Is Ehlers-Danlos syndrome age-specific?
I'm no expert, but as far as I know, no, this is as a consequence of a genetic disease, you will have manifestations of the mutation and the disease with different severity with increasing age, as with all genetic diseases. But as far as I know, it's not a specific age.
Okay, thank you. Any more questions? Nobody? Oh, you will have plenty of time to ask questions also after the next lecture. So if yes, there is one more question coming here. Do it have any chance, it must be risk in cousin marriage? It's still talking about Ehlers-Danlos syndrome.
Yes, with all genetic diseases, it increases, the risk increases with cousin marriage, correct.
Okay. If no more questions, thank you very much, Donald. And then we will be back in approximately 12 minutes for the next lecture. Thank you very much. Welcome back.
Not 10:15 yet. It's not on my computer. Let people finish their coffee.
But now it is.
No, it is.
Yeah, now it is.
Okay, so welcome back, all of you. Now Donald will continue to talk about the integrins, and we look forward to your talk. Please, Donald.
Thank you. Can you see my screen this time?
Yes.
Okay. Okay, so we will continue then. But now I cannot move my video. Oh, now it comes. Right, so what we will do during this lecture is that I will say a few words about history, I will talk about structure, we will talk some repetition from previous lecture about the ligands, and we will say not so much, but some about integrin signaling, And very importantly for this course, where we talk about fibrosis, is the role of integrins in TGF-beta activation. And if we have time, this might not be the case, we will also say some about pathology of integrins, or pathology caused by defects in integrins, or integrin function, I should say. We looked at this slide last time, and we said that the extracellular matrix gives signal to the cell. So a cell can tell, is it in the right place? Is it doing the right thing? Or this is information, the positional information that the extracellular matrix can give. So as I said, when I started as a graduate student in the 1980s, then integrins had not been isolated. So in particular, two scientists contributed to the discovery of the integrin family. So integrins evolutionary have been around for millions of years, but we haven't known them in the scientific community until the 1980s. So the two scientists I would say that were very important for the discovery of the integrin family was Erkki Ruoslahti in La Jolla, California, and Richard Hines in Boston, USA. So the name integrin was given in 1987 by Richard Hines, and to denote a type of membrane protein that's important for the integrity of the cytoskeleton and the extracellular matrix. So it's a link between the outside and the inside. And since then, the number of integrin articles has increased, and now on the average, I think between 50 and 100 articles per week are published on integrins, and in total, now a large number of articles has been published. So I think a zero is lacking in this updated picture. I need to fix this. So what about integrin structure? How are they built up? This is a schematic illustration of one alpha subunit and one beta subunit. So integrins are composed of non-covalently associated alpha and beta that come together. And the alpha subunit we now know is composed of a globular domain, which is composed of a 7-bladed beta propeller. And this is followed by a side domain, a COF1, COF2, a transmembrane, and a short cytoplasmic domain. Whereas the beta subunit has some other domains, including some EGF-like domains. The first integrin to be where the structure was determined at the atomic level was alpha V beta 3 integrins, where scientists worked for 5 years in order to determine the structure. So what one did was to delete the transmembrane part and express the extracellular part, and then one crystallized. The surprising thing when the crystal structure came was it looked like this. It was like a blob, and there wasn't that particular structure, much structure. But what scientists had to do in the computer then was to unfold this structure, and then this is what it looks like. So one important finding from this crystallization was that integrins can assume different conformation. Unfolded inactive and unfolded active conformation. So this particular integrin turns out to bind ligand at the interface between the alpha and beta subunits. So this is, for the majority of integrins, that's the case. Both alpha and beta subunits are needed for ligand binding. But a subset of integrins, actually 9 out of the 18 alpha subunit have an extra domain. It's called the I domain, inserted domain, that sits on top of the beta propeller. And those integrins bind ligands only via the I domain. So the beta subunit in this integrin is not directly involved in ligand binding. And here you see the difference between a bent inactive and an active ligand binding alpha I domain integrin. Here again is another schematic illustration of the difference between an inactive and an active integrin. We said in last lecture that all genes have now been sequenced. We know we have about a little bit more than 20,000 human genes. And we also know how many integrins there are. There are no more integrins to be found. They have all been characterized. And the last integrin has been characterized in my laboratory. It's called the alpha 11 integrin, and it is part of collagen receptor subfamily. So integrins are dimers. They can only, one beta subunit can only bind one alpha subunit, but they're different combinations. So there are 12 different alpha subunits that can combine with beta 1, for example. Some integrins, like the alpha 6 integrin, can bind to either beta 4 subunit, which it does in hemidesmosomes, or beta 1 subunit, which it does in endothelial cells. And some integrins, like the alpha V integrin, can bind to multiple beta subunits, not just two. It can bind to beta 1, beta 3, beta 5, beta 6, and beta 8. So there are different ways to divide integrins into subfamilies. Here is according to ligand. So we have collagen binding, laminin binding, RGD binding, and then here a subset of integrins that are almost exclusively found on leukocytes. Right, so let's have a little repetition from last lecture and go back to the ligands and look more in detail to the ligand binding regions. Again, this is extracellular matrix, integrins. Let's find the laser pen again. Integrins at the cell surface, and here are the different integrin ligands. And when we look at the different matrix components, to varying degree, they form integrin ligands. Probably the integrin ligand that has been studied the most is fibronectin. This is because it's present in the blood at high concentration. It's easy to purify. So fibronectin is a very good integrin ligand. Laminins are produced by some tumors like the EHS tumors, so it's also easy to purify and has been extensively studied. And then there are many other proteins. The more difficult the protein is to purify or produce recombinantly, the less likely it is that it has been studied in the integrin context. Collagens are also integrin ligands. Not all of the 28 collagens bind to integrins. And cells usually have a restricted set of collagen receptors. So it's not as adhesive as fibronectin, as we said, which have many different integrin receptors. Proteoglycans, as we said in last lecture, are very negatively charged, are not usually your prototypic integrin ligands, but integrins can bind to fragments of proteoglycans, and the syndicant proteoglycans can activate integrins, which is a new finding, but they can also act as we said, co-receptors for growth factor receptors. Right, so we said that collagen was the most abundant and it's triple helix. When it comes to the place in collagen where integrins bind. Integrins, in order to bind to collagen, need the triple helix, so denatured collagen, and everybody knows that denatured collagen is gelatin, what we have in candy and in cooking, right? That's denatured collagen. But in order to integrins to bind, the collagen need to be native. And this was a Cambridge scientist called Richard Farndale that in detail studied what region of collagen do integrins bind to. And he identified the sequence GFP-GER as in collagen type 1 as being important for integrin binding. What's very particular about collagen when you compare it to other matrix protein is that it forms highly fibers and fibrils. And on top of that, collagen is coated with many proteins, including proteoglycans like decorin and so on. And on top of that, collagen is very tightly packed. So in the end, there's not that much room for integrins to bind in the mature tissues. And this is what we said, that maybe it's so that in tissues, not so much in mature tissues is available for cells to bind. You need partial proteolysis, or you need the collagen to be freshly synthesized in order for collagen binding integrins to be active. We also talked about laminin, the cruciform molecules in basement membrane. Here it's actually been very difficult to identify the cell binding site. So this is a huge molecule, so it has a molecular weight of about 1 million. And fairly early on, one realized that this region was important, both for binding of another laminin receptor called dystroglycan, which is a non-integrin receptor for laminins, and integrins. And the mapping that has been performed is actually shows that it's the gamma 1 subunit at the end here that's important for integrin binding. We talked about fibronectin, which is this dimer, which has in the 10th fibronectin type 3 repeat has an exposed arginine glycine aspartic acid sequence that is recognized by integrins. And I included this slide just to show that integrins, which are for sure the major group of matrix receptors, can also bind to other cells. So it's a cell-cell receptor. But unlike other cell-cell receptors like cadherins, which keep cells tightly together most of the time, the interaction between integrins and cell surfaces is more transient. So it's when immune cells interact with the vasculature and those type of interactions, not the stable long-term interactions that cadherins are involved in. So this is the cell surface ligands are vascular cell adhesion molecule and intercellular cell adhesion molecules and so on. So this is a summary of that. Here is another division from a review article by Martin Humphreys showing where different integrins are grouped according to the ligands. Here you have collagen binding integrin, here you have laminin binding integrins. And let's see where you have fibronectin. Here you see the many integrins that bind to fibronectin. And here is just a summary of the cell recognition motifs that have been characterized for some of the integrin ligands. When you open a textbook and read about integrins, many textbooks will say that integrins are RGD binding proteins. This is a table I made for a review just to show that this is not true. As we said, there are 24 integrins, and 8 of the 24 integrins actually recognize the RGD sequence. So it's a subset of integrins that recognize the RGD sequence in the ligands, but it's not a general property of integrins. If we want to generalize about the importance of different subunits of integrins, we can say that the beta subunit is by far the integrin subunit where we know the most about its function. We know that the beta subunit is important for the association with the cytoskeleton. And we also know that the beta subunit is important for integrin signaling. For the alpha subunit, we know a little bit. We know that the alpha subunit is a subunit that determines ligand specificity, and which occurs either at the I domain or at a specific region in the seven-bladed beta propeller. But much of what the rest of the alpha subunit does is unknown. So this has been studied to a limited degree, and this is an area of future research. So one picture that is emerging is that the alpha subunit help the beta subunit in signaling and in other functions that previously had been said, oh, this is just a beta subunit, but it's becoming clear that the alpha subunit also contributes to these processes, but this is work in progress. So what do we know about integrin function? Yeah, I said they've been around now for some decades, so we're learning more and more, but we still don't know everything. We know that integrins, when we study them in the laboratory, are important for cell attachment, they're important for cell migration, and through their signaling function, they affect a number of processes in the cells. If you plate a certain cell on either fibronectin or laminin, this can determine how this cell will behave. Will it differentiate or will it de-differentiate, and so on. So it affects cell differentiation, it affects cell proliferation, and by influencing gene expression, it almost can influence anything in the cell. So we know that integrins are important in attaching cells to the matrix, and the type of attachment that integrins make are different depending on the integrin and depending on the matrix. So one type of adhesions that we will look at how you can study it in today's practical part are called focal adhesions. So these are like feet-like structures that the cells form when you grow them on a stiff substrate in vitro. In the body, we're not full of focal adhesion, but similar adhesion points where cells attach with the same molecular composition exist also in our bodies, but we don't necessarily have the same stiffness as on a hard glass or plastic surface, and therefore size and composition varies in the body. We already looked at hemidesmosomes, which is the structures that epithelial and endothelial cells form with basement membrane. Here, it's the intermediate cytoskeleton, intermediate filaments that interact with basement membrane via an adaptive protein called plectin. And the latest addition to this type of adhesions was a discovery made by a Swedish scientist at Karolinska Institute, Staffan Strömblad, who discovered that one particular integrin, alpha V beta 5, which is a vitronectin receptor, keeps cells attached during cell division, and in a special type of adhesions called reticular adhesions. And we will look at the schematic illustration. Here are the feet the cells form in culture that you will, in the practical demonstration today, we will look at how you can prepare cells to look at focal adhesions. Here you see integrins via tailin linked to the actin cytoskeleton. The other type of structure, the hemidesmosome, here you have a specific integrin bind via the protein plectin bound to intermediate filaments. And here is actually zooming in. We've looked in this picture, we look at a few individual focal adhesions. In reality, of course, in the cell, there are many integrins that are clustered, and this is more like what it looks like. And when you look at the nanoscale architecture of focal adhesions, you can divide the protein interactions into different domain, signaling layer, force transduction layers, and so on. Here is an illustration of this paper that was published in Nature Cell Biology in 2018. Here you see a cell which, when it is about to divide, rounds up, and it has these very, very fine, small attachment points, and these are called reticular adhesions, and that's where alpha V beta 5 sits and keeps the cell attached, why in order to have successful cell division. And when beta 5 is lacking, then you don't have a normal cell division, and cells actually fail to divide properly. Okay, those were three types of contact point. When we look at integrins in vivo, they will of course look differently depending on what integrin you look at and what tissue you look at. Here is a picture, a cross section of a skeletal muscle, and here is a picture of the epidermis in skin, where you have alpha 6 beta 4 sitting at the basal keratinocytes. Here is muscle fiber which have alpha 7 beta 1 in their sarcolemma. Right. So maybe you're already guessing that integrins, which are kind of important, kind of present in varying numbers and various type in every cell in the body, except for erythrocytes, that they have a lot of interaction, and that is indeed the case. So here is a map of what's called the integrin adosome. So this is showing different proteins that interact with integrins, here showing intracellular interaction. Okay, so then we move on to signaling. And when you talk about signaling over the membrane, there are different ways that signaling can be transmitted from the outside to the inside. And if we talk about tyrosine kinases, whether a tyrosine kinase will transmit a signal will depend on, for example, how dimers are stabilized, as in this case. When a ligand bind, you have a stabilized receptor that can signal. Integrins are involved in two types of signaling. When the signal comes from the inside, it's called inside-out signaling, and when the signal comes from the outside, it's outside-in signaling. And both of these types of signaling are transmitted across the membrane via conformational changes. And we said that much of what's known about integrin function comes from studies of the beta subunit. And this is a slide showing the type of interactions that the cytoplasmic tail of the beta subunit has. And here you can see that two crucial proteins important for activating are tailin and kindlin. And both tailin and kindlin are involved in integrin activation. That is taking the integrin from this stage to this stage. And how do they do this? Well, tailin binds to the what's called the membrane proximal conserved NPXY motif of the beta subunit, and this leads to integrin activation. And tailin itself is actually inactive and needs to be activated, and that can occur by small GTPases in the cells, here exemplified by RAP1 and RIAM. And this is the same slide showing how these small GTPases activate tailin, so tailin can bind to the beta subunit of the integrin, and this completes what's called inside out. Inside out. Once the integrin is active, it can involve in outside in signaling. Here is a closer look at how tailin works by binding to this membrane proximal part. So tailin is activated and then binds to this NPXY motif in the proximal part of the beta subunit. Here again is membrane proximal, membrane distal NPXY motifs. So tailin bind here and kindlin actually bound to this membrane distal NPXY motif. How tailin and kindlin collaborate, and the details of this is not known, but it has been suggested that they collaborate in some way, but as I said, exactly how is not known. One of the most recent findings during the last few years is that kindlin might also have a unique function. It might cluster several integrin together by binding to one beta subunit and then to the other beta subunit, so have a clustering function. So one can ask oneself, why is it important that integrin activity is activated? Wouldn't it be better for the cell just to produce an active integrin, and once it has done its function, it can be degraded and behave like normal membrane proteins that are produced, degraded, and so on. But the reason is actually that integrins have important function on some cells in the body. And the two prime examples where integrin activation is absolutely crucial is on during blood coagulation and inflammation. So on platelets, there is one particular integrin alpha 2b beta 3 that binds to fibrinogen and takes part in clot formation. And in case you don't have an injury and your platelets are not going to be aggregated, this integrin is 100% inactive. If this integrin was kind of existed in a little bit of active or so, this would cause to blood clots forming throughout the body. So in order for this integrin to work, it's absolutely required that when platelets are inactive, so are the integrins. So there is no aggregation taking place unless you have activation. The other instance where integrin activation is important is during inflammation. When leukocytes, which in order to go into the site of inflammation, first need to bind to the vascular endothelium and extravasate. But this only happens when you have an inflammation, and this only happens when the integrin is activated and it can bind to the endothelial cells. If immune cell integrins were active all the time, we would have immune cells roaming throughout our bodies, which would be a very non-economical way for the immune system, where it couldn't concentrate its force on exactly where it's needed. And I think this is just detailed pictures of how integrin becomes activated on a leukocyte, a white blood cells. That integrin exists as an inactive molecule when there are activation signals, tailin binds, and via inside-out signaling, integrin becomes active and can bind to ligand on opposing cell. So one of the puzzling things when integrins had been purified, which was a great thing because before one didn't believe that there were matrix receptor. All of a sudden one had a big family of 24 members. But what one didn't understand, how can this receptor be so important when it has such a tiny, short little tail? And unlike tyrosine kinases, which have enzymatic activity, these short tails cannot have enzymatic activity. So the problem was solved when one found that integrins associate with a soluble tyrosine kinase called focal adhesion kinase. And this soluble tyrosine kinase can generate a lot of signals that go into the cells. So focal adhesion signals, outside in signals, are central. Focal adhesion kinase is very central for integrin signaling. Focal adhesion kinase, as I said, is a soluble tyrosine kinase. We don't have time to go into the different domains. But this is also an auto-inhibited kinase that can be activated, and when it becomes activated, it becomes autophosphorylated on tyrosine 397. And this leads to the association with SRC, which is another tyrosine kinase that continues then together with focal adhesion kinase to phosphorylate downstream targets. And here is a more detailed schematic illustration. The integrin binds to the activated focal adhesion kinase, leads to association of SRC and further activation. So downstream of focal adhesion kinase, there are many different signaling pathways. One major downstream signaling pathways are the mitogen activated protein kinases, MAP kinases. But integrins have also been reported to signal without focal adhesion kinase. Some of these studies, which were FAK independent, have not stood the test of time, and I'm not actually so sure, but it's clear that the most common signaling via integrins is dependent on focal adhesion kinase. And when we're talking about MAP kinases, there are above three that the three that we in general talk about is P38, JNK, and ERK. These are P38, JNK, and ERK are the three major MAP kinases activated by integrins. So this leads to signal that leads to transcription factors going to the nucleus, affecting also transcription. Another set of signals are focused on the cytoskeleton, where three small GTPases, CDC42, RAC, and RHO, all have different effects in the cells. CDC42 is involved in filipodia formation, RAC in lamellipodia formation, and RHO in stress fiber formation. And these small GTPases are all regulated via integrin via focal adhesion kinase. And so there are different type of interactions that occurs in, and it's different factors in different cell types. So this is a very finely tuned signaling mechanism. So you think about a simple mechanism. How does the cell crawl? And you start realizing when you look at the molecular mechanism, it's extremely complex. You can spend your life investigating how a cell crawls because the molecular machinery and the signaling function are so complex. This is an old slide showing the activity of just these three GTPases in a cell where RAC and CDC42 are active at the front of the cells, whereas at the back of the cells where processes has to be activated so that the cells come loose and can move forward, Rho A is active. And in the central part of the cell, of course, Rho is active to generate the stable feet, the stable focal adhesions that exist there. So this is just to illustrate that cell migration is a complex process and integrins have a central part of this. So I think the last thing that we do is talk about something that's super important for this course when you talk about, and also for the DQ course, that is how do integrins regulate matrix turnover. And it turns out that central growth factor cytokine for this is transforming growth factor beta, TGF beta, which is synthesized in a complex manner in a complex. And the actual growth factor, TGF beta, is secreted bound to what's called latency associated peptide. That's the stuff here. It's non-covalently associated with LAP. So TGF beta signaling occurs via binding of TGF beta, active TGF beta to receptors, leading to, in the canonical signaling pathway, to recruitment of what's called SMAD proteins that then go to the nucleus to affect transcription. And what integrins do is that they activate TGF beta bound to this latency associated peptide, LAP. And there is a specific subset of integrins that do this. And these are the integrins that contain the alpha V subunit. And the first alpha V integrin shown to be involved in this was alpha V beta 6. And this was a super high profile paper showing that if you subjected mice to fibrosis, lung fibrosis, and they lacked beta 6, they were protected. And this was, one could see that this particular integrin was needed in order to activate TGF beta. And since then, a number of other integrins have then been shown to be important. And the latest one, where much of the interest is right now, is alpha V beta 1, which seems to be central to fibrosis on fibroblast and cancer-associated fibroblast. So the way that integrins work to activate TGF beta here is from the classical experiment where the epithelial cells express alpha V beta 6. It binds to an RGD sequence in the latency associated peptide, opens up the complex and frees active TGF beta. What has recently been shown is that the stiffness of a tissue affects the efficiency of this activation. So if you have a stiff tissue, like maybe fibrotic tissue, that means that it will be easier to activate TGF beta. It has been kind of pre-activated, less strain is needed to activate. And here again is close up how the integrin, via being connected to the cytoskeleton, opens up the LAP, so TGF beta is released. And the final confirmation that this is indeed the case came with a crystal structure from Timothy Springer's lab a few years ago, showing that TGF beta is hidden within LAP, and when you open it up, it is freed from this. So the relationship between TGF beta and stiffness is like the stiffness is the counter force to the straight jacket that TGF beta is in when it is produced. So this is another example of how important alpha V integrins are for TGF beta activation. So this is an example of liver fibrosis where alpha V beta 1 on the stellate cells is involved in activating TGF beta. So if you induce fibrosis and include an inhibitor against alpha V beta 1 integrin, then you have no fibrosis because TGF beta is not activated. And actually, the remarkable finding was that if you knock out integrins involved in TGF-beta activation, you have a phenotype that's reminiscent of mice that lack TGF-beta. So to summarize, integrins are mechanical links and has crucial function in some tissues like the skin and muscle, where structural integrity is important. Integrin function is important in particular in blood coagulation and immune system when we talk about integrin activation. Those are two prime examples where regulating integrin activity is important for integrin function. And a lot of functions related to integrins has to do with their signaling function. But then it's important to remember that integrins are just one type of signaling receptor which integrates with other signaling receptors, and together they work to affect functions in the cells. And almost 3 decades after integrins were discovered, there are many, many questions related to integrin functions that remains to be learned. So we didn't have time now to go through disease. Maybe I'll just flash through the slides that integrins are important for muscle attachment in flies. There is immune disease where immune cells are not activated properly. There is a bleeding disorder where integrins are not functional. We talked about epidermolysis bullosa with laminin. There are also integrin mutations that gives the same phenotype. And most integrins have been knocked out with various phenotypes. Some have severe phenotypes, some have very mild phenotypes. In dogs, a particular integrin is important for cartilage development. These dogs get a chondrodysplasia with a disease of the paws. And you can use antibodies to integrins to inhibit immune cells in different diseases. And the last slide shows the importance of integrins in tumors where they are involved in a number of cells, both immune cells, cancer-associated fibroblasts, and on the tumor cells themselves. So I think I stop there. Thank you.
Thank you. Thank you very much, Donna, for a nice lecture. I see that we have one question here, and two. The first one. Thank you for
so people
a
are
very
not
informative and educative lecture. Could you please give any comments of the turnover or assembly, disassembly of adhesion complexes during cell cycle process, especially from G2 to M and then from G0 to G1? In the mitosis phase, are there any other integrins that contribute to cell attachment? There's several questions here. If lack of a reticular adhesion, it could cause cell cycle arrest, right? And how? So if we start with the first question, could you please give any comments of the turnover or disassembly of adhesion complexes during cell cycle process?
No, I have no information whatsoever of that. I'm sure somebody has studied it. I have no idea. But the puzzling thing that was only solved in 2018, that was how do cells remain attached during the rounding up? in the cell cycle, and that was solved with this finding with alpha V beta 5. As far as I know, that's the only integrin that keeps the cells attached in vitro. How that corresponds to the in vivo situation during cell division is of course unknown as far as I know.
I will read the following questions, but
Can
please.
can we not ask people to ask questions directly, not having to
That's
go with?
what I was going to do. You can please also use the raise hand to ask the questions directly instead of using this chat function. So another question is loss of attachment for adhesive cells. Do they detach and just die?
Yeah, I mean, most cells need attachment, otherwise they undergo apoptosis, right? So preventing detachment is actually an active process. So it's not a passive process. So you need some signaling to keep the attachment to prevent apoptosis, if I understand it correctly.
Okay. I have got any raised hands. There are several questions here. You mentioned integrins underlie focal adhesion complex creation, which are accentuated on stiff tissue culture plastic. What are integrins binding to on the plastic? Are they pressure or stiffness sensing?
Well, integrins are binding to either proteins that they make themselves in the culture, but most often fibronectin, as we said, is present in serum in high concentration. We said that 200 microgram per ml in normal serum, bovine and human. And if you have a 10% serum, that means you have 20 microgram per ml, which is about 20 times above the saturating concentrations. So you have very high concentration of fibronectin that cells are binding to. And as you say, the plastic is a very stiff surface, non-physiological, so you create an accentuated contact point that normally doesn't exist in vivo, where you have much lower stiffness.
And then Ratika is wondering, how is alpha SMA regulated by TGF beta? Is it acute or a chronic effect?
You cannot answer in a generalized manner. So TGF beta has a very stimulatory effect on many things in cells, but that depends on the dynamics of the cell, what cell type, and TGF beta signaling, because you have both canonical signaling, which works via transcription, via SMADs, that of course will take longer time before you have an effect, but it also works via non-canonical signaling, which is typically MAP kinase activated. That is immediate and can have more rapid effects.
Okay, thank you. We have a one raised hand, but I will take one more question here from Tilini. Thanks for a nice lecture. Did the COVID vaccine related blood clots also have integrin activation?
They must have, because otherwise coagulation wouldn't have happened. So in order for coagulation to work, fibrinogen has to bind, and that occurs via integrins. So whether integrins activation is affected, that's another issue, but integrins are certainly involved in all types of coagulation on involving thrombocytes, since they are the major fibrinogen receptor. Or
I thought we had a raised hand here.
There's another question and answer. I don't know if it's checked. Are there
Yeah.
any situations where the integrin subunits expressed by the cell changes?
Yeah.
Yeah, all the time. So integrin synthesis is very dynamically regulated. So this happens all the time.
So, are there any more questions? Yeah.
Maybe we can encourage to use the raise hand function. It would be very nice for us to see you also. So please use the raise hand function when you ask questions to as much as possible. Thank you.
Thank you. No more questions. So then, thank you very much. And please remember to use the raised hands next time.
What happens now, Marion?
Sorry?